हेलो बच्चों, मैं हूं आपका केमिस्ट्री वाला और हम आज पढ़ने वाले हैं बायोलॉजी। आप सोच सकते हैं मैं केमिस्ट्री वाला नाम रखकर बायोलॉजी क्यों पढ़ा रहा हूं। क्योंकि मैं सिर्फ और सिर्फ केमिस्ट्री नहीं पढ़ाता, मैं बायोलॉजी भी पढ़ा सकता हूं। अब हम बायोलॉजी पढ़ेंगे। हम सेल द यूनिट ऑफ लाइफ से माइटोकॉन्ड्रिया और इसके बारे में आज आप पढ़ने वाले हैं। Mitochondria. Mitochondria were first discovered by Kolliker in the muscle cell. Remember this point, people. Kolliker in the muscle cells. Altman named them as bioplasts. The term mitochondria was coined by Benda. Who coined the term mitochondria? Benda. They are present in all eukaryotic cells. The number is more in animal cells than in plant cells. So, in plants, there are fewer mitochondria than animal cells. The number of mitochondria are more in meristematic cells than permanent cells. The diameter of mitochondria ranges from 0.2 to 1 micrometer and 1 to 4.1 micrometer in length. The ultrastructure of mitochondria. As you can see the diagram of mitochondria, you can see the outer membrane, inner membrane, the matrix, the cristae, the inner space. Mitochondria is a double membrane organelle. As you can see, the outer membrane envelopes the inner membrane. The inner membrane has cristae, has infoldings called cristae. Mitochondria is surrounded by double membrane envelope. The outer and inner membrane are separated by perimitochondrial spaces. Usually, the outer membrane is smooth and the inner membrane forms tubular infoldings or invaginations in the matrix, known as cristae. As I told, the cristae increases the surface area. The inner membrane contains respiratory chain. The average number of oxysomes in each mitochondria is 10 power 4, 10 power 5. Each oxysome consists of a stalk like F0 and F1 head. F0 is the stalk and F1 is the head. Or you might be wondering, oxysomes, what are oxysomes? These are like for ATP synthesis. F0, F1. Let me show you. Where is this? Oxysomes are like something like this. This is the membrane. This is the stalk. This is the head. When H plus ion passes from higher concentration to lower concentration, it releases energy. That energy is converted to ATP. This is called ATP. This is how ATP synthesis in mitochondria occurs. The matrix of mitochondria contains a single naked double-stranded DNA. All three types of RNA are present in mitochondria. See, the mitochondria has its own DNA, so it can exist independently also. So most of them consist as so they are also called semi-autonomous cell organelles because they are capable of self-duplicating. Both transcription and translation takes place in mitochondria. Types of RNA present in mitochondria: mRNA, rRNA, tRNA. The functions of mitochondria in eukaryotic cells, except glycolysis, which is the first stage of the ATP synthesis, the remaining steps of aerobic respiration occurs in mitochondria. Link reaction. 1 second. Okay guys, as I was saying, in eukaryotic cells, except glycolysis, the remaining steps of the aerobic respiration occurs in mitochondria. Link reaction and Krebs cycles takes place in mitochondria. Electron transport through the oxysomes and oxidative phosphorylation takes place on inner membrane of mitochondria. They convert potential energy into kinetic energy. Remember this point, it has been asked in NEET many times. Mitochondria do what? They convert which energy into which? They convert the potential energy to a kinetic energy and store the energy in the form of ATP, adenosine triphosphate, which is the chemical energy of the cell. Hence, mitochondria is also called bifurnaces because they produce ATP, or they are also called as powerhouse of cells. ATP molecules are also called energy currency of the cell. Ribosomes. Now we move on to another topic, which is called, no, yes, we move on to ribosomes. Ribosomes are granular structures first observed under the electron microscopes as dense particles by George Palladay. They are composed of ribonucleic acids, RNA, and proteins. They are not surrounded by any membrane. Eukaryotic ribosomes are 80S, while prokaryotic ribosomes are 70S. So, ribosomes occur in both eukaryotes and prokaryotes, which means ribosome is the only cell organelle which occurs in both prokaryotes and eukaryotes. Hence, S stands for Swedberg constant or unit, Swedberg unit, segmentation coefficient. It is indirectly measures of density and size. Both type of ribosomes are composed of larger, smaller subunits. As the 70S ribosome, it contains 50S, a bigger subunit, and a 30S, smaller one. And for the 80S ribosome, which is present in eukaryotes, except plastids and mitochondria, they contain 60S and 40S. The two subunits in both types of ribosome associates with each other in presence of magnesium ions. So this is really important here. Seriously, I'm gonna mark this point. Seriously. The two subunits in both types of ribosome associate with each other in the presence of magnesium ion. This is a big important. Keep this. This they're gonna ask in NEET exam. They asked the NEET exam many times over. So you see the 50S and 30S or the 60S and 40S, they come together and form 70S and 80S ribosomes when in the presence of magnesium ions. If there's no presence of magnesium ion, they remain separated. So yeah, this is really important. Next, ribosome provides space and raise and as enzyme for synthesis of proteins. You all know this in ribosome synthesis proteins by translation transfer. Cytoskeleton, an elaborate network of filamentous proteinous structures present in the cytoplasm are called collectively referred to as cytoskeleton. Eukaryotes. One second, guys. There have been some disturbance in my surroundings. Let me take some dihydrogen oxide. Okay. Eukaryotic cells contain three major components of cytoskeleton: microfilaments, intermediate filaments, and microtubules. The cytoskeleton in a cell is involved in many functions such as mechanical support, maintenance of cell shape, cell motility, intercellular transport, signaling across cell and karyokinesis, movement of chromosome during division, and etc. They generally form the structure to the cell. Next we move on to Cillian and flagella. I think we have a separate thing for this. Okay, the video picture didn't come out right. You have to adjust for this one, guys. Cillian and flagella. Cilia and flagella. Cilia and flagella are filamentous, flexible processes of cytoplasm, which act as organs of locomotion. Both of them are identical structures, but they differ in their number, size, and function. Ultrastructure of flagellum and cilium. Flagella are locomotory organelles of the mastidopharon protozoans. You get this right? They are only present in protozoans, means they are not present in other organisms. Flagellum consists of a long, stiff, axial filament called axoneme that arises from the basal granule. The axial filament or axoneme shows 9+2 organization under the electron microscope. The two central singlets, each with one microtubule, are enclosed by fibrous inner sheath. 9 peripheral doublets form a cylinder between inner sheath and outer sheath. So I'm just gonna draw the structure of cilium here. The structure of cilium and flagella are almost same, so you can understand, but only there's only one difference in them. This is outer sheath. Here they contain tablets. Sorry for the drawing. Oh my god, this thing. I'm not a good drawer. I'm not a good artist, you know. Okay, that came right now. Okay, as you see, there are 9 things. Okay, I know that sucks, but consider them as tablets. Okay, now I'm getting. Oh no, I lost. Come on. 1, 2, 3, 4. There should be no space between them. They're called A and B. You know, they should not interlinking each other. Yeah, 1, 2, 3, 4, 5, 6, 7, 8, 9. Oh, I'm pretty good, I guess. I'm not a good artist. They have radial spokes coming from them, and they stop here. Yeah, there. Radial spokes. I'm not drawing circles. I know you get it. Come on. Yeah. Now this is the inner sheath, and here we have 2 separate central microtubules, and they are connected by a filament like this. They're not attached to each other. And here are some dynams coming out from the B thing. 1, 2, 3. 1, 2, 3. Not only 3, there can be more. Yeah, it occurs for everything. And there's an interconnecting bridge between every doublet. Man, my drawing's not good. Yeah, but that's it. This is called Nexon. Made up of Nexon. Come on, Nexon. Yeah, that's really not good, but bear with me, guys. This is how it should look. Now, this is the arrangement of flagellum. And for cilium, there's nothing. They don't have these central tubules. That's it. So in flagellum, we have 9 plus 2. And in cilia, we have 9 plus 0. That means we have no central microtubules. That's the only difference between cilia and flagellum. Everything is same between them. Functions are similar. Now, cilia are short hair-like structures present all over the surface of the body or confined to specific regions of the ciliate protozoans. Primitive forms like holotrix parazoans have cilia all over the body surface. In Peritrichus verticella, peristominal cilia are seen. Sucterians like Acineta bear cilia in young stages. So this is a brief review of cilia, flagella, mitochondria, ribosomes. Now we're going to move to one of the most important topics of anything, that is the nucleus. Here's centrosome and centriole. They're almost not that tough. See this structure. See centrobule mitochondria. And similarly, this thing structure is almost same, but has triplets and a single central hub without that's not like this hub. Okay, we have to read this thing. Okay, let's do centrosomes and centrioles first. Centrosomes and centrioles. Centrosome was discovered by Boveri. Centriole, the 2 components of one centrosome, are seen arranged at right angles to each other. So guys, not like they're parallel to each other, they are arranged right angles to each other. Like one is going to be this Wait, I'm gonna show you. And again, this thing. Not a good. This again. One is like this. The other is like this. Hope you get what I'm saying. Yeah, and like that. So the two are there, and one's gonna be like that, one's gonna be like that. And they are surrounded by pericentriolar materials. Centrum, center, are non-membranous cell organelles which are seen in pairs. A pair of centrioles is called diplosomes. So a pair of centrioles means two centrioles are called as diplosomes. Diplos means double. Soma means body, means double body. You get it? Centrioles are seen in all animals except mammalian erythrocytes because they in mammalian erythrocyte they get rid of all the organelles mostly. They don't have nucleus because they have to have more amounts of hemoglobin for the exchange of gases. So it removes the centriole, gives more space for the hemoglobin. So mammalian erythrocytes do not have centrioles, and centrioles are not even found in primitive fungi, protists, bryophytes, and pteridophytes. Multiple centrioles are seen in bone marrow cells. It is made up of a protein tubulin, a small amount of ATPase. So what is a centriole made up of? A protein tubulin and a small amount of ATPase. Ultrastructure of centrioles. Centriole has cartwheel-like appearance due to presence of spokes, hub, and peripheral fibers. The part of centriole are as follows: microcylinder, a hollow tube-like structure which is open at both the ends, forms the microcylinder, which is membraneless. At the peripheral portion of the microcylinder, 9 triplets are seen running parallel and longitudinal to each other. Other. The angle between the fibrils is 40 degrees. Triplet fibers. The 9 triplet fibers are seen in central parts do not have any fibrils. Therefore, centriole shows 9 plus 0 condition. The 3 microtubules are joined and they share a common wall matrix. Space inside the microcylinder or centriole is called matrix. Outer part is dense compared to the inner part. The hub, rod-shaped proteinous structure that runs throughout central part of the centriole. Linkers, proteinous connections seen in between triplet fibers. So in flagella we have doublet fibers, and in centrioles we have triplet fibers. So here, now instead of 2 we will have 3. And the poke, the proteinaceous strand seen radiating from the center of the hub is called a spoke. Spoke connection. They are strands that connect CA linkers with radial spokes. Macioles. They are 9 in number, seen outside the matrix. They are not static. They function as nucleating centers for formation of astrial rays and more centrioles. So this macioles is an important thing you have to learn. So the macioles are like the 3 things. They're 9 in number. They're seen like outside of the matrix. They are not locomotory things because they're not static. They're not static. And they help in formation of astral rays, which is helpful for forming a more centrioles and helpful for cell division. Now finally we have to move on nucleus. Come on, nucleus. Yes. Hey guys, nucleus. Most thing. Nucleus is a conspicuous spherical structure which controls and coordinates various life activities of a cell. It is the largest and most dynamic cell organelle. It was discovered by Robert Gallen orchard root cells. Later, the materials of the nucleus stained by basic dyes was given the name chromatium by Fleming. It is present in all eukaryotic cells except mature RBCs and sleeve elements of plants. J. Hammerling conducted experiments on green algae, Acylobacterium, Acylobularius, and proved hereditary role of nucleus based on the number of nuclei present in cells. Also of 3 types: uninucleated or monokaryote, These are most common type of cells with single nucleus. Binucleated or dikaryote cells with two nucleus. Example: Paramecium, members of Astigomytonia, Basidomycotonia, tapered cells of Anthropods, Coventocytic or multinucleated or syncytium. This is important. Cells with more than two nucleus. Vaucheria algae, Mucorrhizopus fungi, tapered cells of angiosperms. It is the largest cell organelle and occupies 15-20% cell mass. Size of nucleus of cell may vary from size to cell to cell. In measurement cells, it is centrally located. In permanent cells, the nucleus lies peripheral and cytoplasm due to the presence of large central vacuole. Shape of nucleus is related to shape of cell. In most other cells, its shape is spherical. In central cylindrical cells, it is elliptical. In human neutrophils, it is trilobed. Structure: the nucleus has four parts. Nuclear envelope: it is a double membranous envelope covering nucleus. It is also called karyotheca or nucleolemma. Each unit of membrane is 5 to 10 nanometers in thickness. The outer layer is called ectokaryotheca or outer nuclear membrane, and the inner layer is called endokaryotheca or inner nuclear membrane. The two membranes are separated by perinuclear space. Outer nuclear membrane is continuous with endoplasm reticulum. Nuclear envelope bears number of perforations and nuclear pores. These are circular with diameter of 10 to 100 nanometers. The nuclear pore may be plunged. Here, see the structure of nucleus. The nuclear pore may be plunged by a large central cartwheel-like diaphragm known as annulus. The total number of pores in nuclear envelope may vary from 100 to 10 to the power 7. Various molecules and substances are exchanged between cytoplasm and nucleoplasm through nuclear pore. Nucleoplasm. Here, nucleoplasm. The nucleus is filled with transparent granular homogeneous fluid known as nucleoplasm or karyoplasm. It is slightly acidic in nature. It is also called nuclear sap or karyola. It is composed of mainly nucleic acids, ribonucleoproteins, glycoproteins, enzymes like DNA polymerase, RNA polymerase, NAD synthase, dipeptide, and it contains nucleus and chromatin reticulum. The nucleoplasm contains many thread-like coiled elongated structures called chromatin or chromatin reticulum. Generally, chromatin reticulum is observed only in the interface of nucleus. So if you want to see a chromatin, you have to see it in the interface of cell division. During cell division, chromatin condenses into thick, rod-like structures called DNA. Chromosomes. Chemically, chromatin consists of DNA, proteins, traces of RNA, histone proteins, and constituents of chromatin of all eukaryotes. Chromatin devoid of histone proteins is found in prokaryotes. Histone proteins have high content of amino acids like arginine and lysine. RNA polymerase is a major enzyme which is non-histone protein. When the chromatin is stained with basic dyes like acetocarmine or fuligen, two types of regions like acetocarmine or fuligen, two types of regions are observed which are known as euchromatin and heterochromatin. Now this is important. Listen carefully. The two types of chromatin, euchromatin and heterochromatin. Euchromatin is active enzymes. It forms loose coiling of nucleosome strands. It is lightly stained fibrous part of the chromatin. It forms the bulk of the chromatin and it contains active genes. Heterochromatin, darkly stained granular parts of the chromatin is called heterochromatin. This term was coined by Emil Hertz. It contains highly coiled DNA strands and it consists of inactive enzymes. Nucleolus. It is a membraneless, darkly stained, round structure present in the nucleoplasm, also known as plasmosome. So what is nucleus or nucleolus also known as? It's also known as plasmosomes. Where are we now? Here, yeah. Nucleolus was first observed by Fontana. Nucleolus develops from nuclear organism, which is the secondary Nucleus develops from nuclear organism, which is the secondary constitution of the nuclear organism chromosome. It contains central. Okay, yes, see, the secondary construction. The chromosome has a primary construction, a secondary construction. Every chromosome has a primary construction, but not every chromosome has a secondary construction. We will see about this later. And remember this: the nucleolus is formed by the secondary construction of a part called NOR. Okay, now the functions. The nucleolus, they produce ribosomes. Hence, nucleus also known as ribosomal factory. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cell activity carrying out protein synthesis. Nucleolus disappears during prophase and reappears during telophase, during cell division. Functions. Functions of nucleus. It coordinates the function of cells or cell organelles. It is also known as cell brain or dynamic center of the cell. It helps in transfer of genetic information from parental cells to daughter cells and from one generation to another generation. It plays an important role in reproduction in unicellular organisms. And now another important topic known as chromosomes. Chromosomes. Hofmeister observed chromosomes and pollen mother cells of Transcandia. The name of chromosome was coined by Walter. Chromum is color and somum is body for darkly stained regions of the nucleus. The chromatin material undergo condensation to form chromosome during cell division. The chromatin contains DNA and some basic proteins called histones. Some non-histone proteins are also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 46 chromosomes. So you may think, a 2 meter long DNA, how is it packed in such a tiny cell? We will see about this. This is also called. They are linked by histones. So we'll learn about this. Chromosomal theory of inheritance was proposed by Sutton and Broveri. Robin Fulgen demonstrated a color test known as fusion reaction of a DNA and showed that chromosomes contain DNA. Walter Fleming introduced the term chromatin to describe threads-like materials of nucleus that became intensely colored after staining. Chromosomes are vehicles of heredity, hence they are also known as physical base of heredity. Number. The number of chromosomes is constant for a particular species. The suffix ploid refers to set of chromosomes. There are two types of ploids, a diploid and a haploid. Diploid means 2 and means there are two sets of chromosomes, and haploid means and they have only a single set of chromosomes. In human, n is equals 23, and the total number of chromosomes in the human is 46. Morphology and the humans are diploid. Morphology of chromosomes. Size is usually measured in mitotic phase because they appear short. Because they appear in mitotic phase. They may be short as 0.25 micrometers in fungi and birds, or as long as 30 micrometers in Trillium, a monocot which belongs to the Liliaceae family, but majority of the metaphase chromosomes fall in range of 3 nanometers to 3 micrometers to 12 micrometers in various organisms. Plant chromosomes are larger than animal chromosomes. Larger chromosomes are Lambrust chromosomes seen in vertebrate oocytes and polythene chromosomes of certain dipteran insects. Wow. So these are important. Lambrust chromosomes and polythene chromosomes. We'll discuss about them in another lecture because we have time for them. Shape. In prokaryotes, it is a circular or ring shape. In eukaryotes, during metaphase, all chromosomes are rod shaped, but in anaphase, they get V, L, J, and I shapes based on centromere position. Best stage study the structure of chromosome is metaphase due to maximum condensation. Chromatid. The longitudinal half of the chromosome is called chromatin. Each chromatin consists of DNA molecules. Metaphase chromosome contains 2 chromatin which are attached to centromere. Chromatonemia. Appearance of chromosomal materials as filaments during mitotic prophase are called chromatonemia. Chromatonemia represents a chromatin in early stage of condensation. Hence, chromatid and chromatonemia are two names of the same. So, chromatonemia and chromatin are same structure. i.e. a single linear DNA molecule associated with histone proteins. Thus, chromatid or chromonemia forms a gene-bearing portion of the chromosome. So, the chromatonemia forms a gene-bearing portion of the chromosome. Centromere. Chromatonemia of the chromosome are connected at the centromere region. Centromere is also called primary construction region of the chromosome, like I said. So I need a diagram to show this. Now I will find the diagram here. You want me to write the diagram, draw a diagram? I think can't bear this. Okay, guys, I know you guys are not that big a fan of my drawings, but have to bear with me. You don't have anything, so. Oh God, I can just see this. This. Okay, got it. Yes. Come on, yeah. That's it. Okay, I got this. Oh shit. So this is the centromere. This they have two kind of distance. This is like the thing. This is the telosome. This is secondary construction I'm talking about. This is the NOR. This is also secondary construction. So we got two secondary constructions and one primary construction. This is a structure of an ideal chromosome. Kinetochore. This shape structures which are made up of proteins seen surrounding the centromere. During cell division, the spindle fibers are implanted to kinetochore discs. So the top portion, like I said, telomere and satellite, secondary construction, chromatid fiber, primary construction, centromere, kinetochore, heterochromatin, euchromatin. Secondary construction. The non-stable constructions at a point of chromosome besides primary constructions are known as secondary constructions. These are useful in identifying particular chromosomes in a set because their position is specific. However, they differ from primary construction lacking of kinetochore. Chromosomes do not bend at secondary construction regions during anaphase, nuclear organization. So as I said, nucleolus develops from the nucleolar organization of the secondary construction. Now we're going to study about nucleolar organizer. These are specific secondary constructions which include the formation of nucleoli. That's it. Satellite. Sometimes the chromosomes bear round, elongated, knob-like structures beyond the secondary construction, which is known as a satellite or tribent. Okay. What is the other name of satellite? It's also called tribent. Trebent. T-R-E-B-E-N-T. Such chromosomes are termed as SAT chromosomes. The function of satellite, there's no particular function of satellite, it's just there for the, there's no particular function of satellite, okay? Telomere, end of chromosomes having polarity are called as telomere. Telomeres prevent the chromosome from fusing between. So telomere plays another important role. You see here. These are the hands of the chromosome. Yeah. So this here we have two telomeres of opposite polarity. Same polarity, and they repel each other. So they get away from each other. If they did not have telomeres, they would eventually be like this. See, they'll fuse with each other. This thing will become this. This should not happen. If this happens, you're aging. There's also an enzyme which prevents this. So this is how a chromosome should look. Without telomere, every chromosome will look like this. This is not good. So that's why people always have chromosomes like that. And the chromosomes will lose their individuality and etc. Classification of chromosomes based on function. Chromosomes are divided into autosomes and allosomes. Autosomes. The autosomes chromosomes which are not involved in sex determination, also called autosomes. Autosomes determine the vegetative characters of an organisms. Autosomes exceed number than allosomes of a diploid component of an organism. Allosomes. These are the chromosomes involved in sex determination and determination of sexual characters of an organism. So there are two types of chromosomes. Get it? One is autosomes and another one is allosomes. Autosomes are like the chromosomes which are not involved in sex determination, Allosomes, which are involved in sex development, with allosome chromosomes are X and Y. There are two types. They are mainly involved in sex determination. If the baby is X and X, we will have a female. If baby is an X and Y, we will have a male. So understand in human there are 22 pairs of autosomes and 1 pair of allosomes. Based on number of centromeres, chromosomes are divided into monocentric, dicentric, polycentric. Monocentric, which have 1 centromere, most common. Dicentric, they have 2 centromeres. Polycentric, they have more than 2 centromeres. And now, based on position, they are divided into telocentric, acrocentric, submerocentric, metacentric. Metacentric, they are perfect. Centromere is located in the center of the chromosome, forming 2 equal arms. These chromosomes appear in V shape during anaphase because they bend to each other. Right? Submetacentric. Centromere is located slightly away from the center and thus forming 2 unequal arms. They appear as L-shaped during anaphase. Acrocentric. Centromere located close to one end of the chromosome and forms 2 arms. They appear as J-shaped during anaphase. Telocentric. They are centrally located at one end of the chromosome. They appear as single I-shaped during anaphase. So guys, I want you to remember the shapes. What is metaphase? Is V shape. This V shape, this L shape, J shape, and the I shape. So remember the shapes. They're really important. They're gonna be asked. These shapes have been asked many times in the karyotype, a group of characteristics that identifies a diploid set of chromosomes is called a karyotype. It represents all characteristics of chromosome seen in species such as number, size, shape, length of arm, relative position of centromere, secondary constriction, and satellites. Uses of karyotypes. If karyotypes of two different species have similarities, it is presumed to represent evolutionary relationship. Karyotypes are just advanced and primitive features of an organism. Ideogram. Diagrammatic representation of karyotype is called ideogram. Ideogram is prepared for diploid sets of chromosome in which pairs of homologies are ordered in series of decreasing size. Nucleosome model. This is a nucleosome model. It was proposed by R.D. Kornberg and J.O. Thompson. It is universally accepted model. According to this model, the chromatin is composed of a string of repeating beta units called nucleosomes. The term nucleosome was coined by Ouellet et al. Nucleosome is oblate or disc-shaped particle with a diameter of 11 nanometer and 5.5 nanometer in height. It consists of 2 copies of 4 nucleosome histones, H2A, H2B, H3, H4. Histone octamer forms a core around which the dsDNA helix is wrapped twice with 146 BP. The nucleosomes are linked together by DNA molecules and linker DNA, internucleosomal DNA, spacer DNA. These are the different names of linker DNA. Remember them. Linker DNA also called as spacer DNA, internucleosomal DNA. It is generally 54 BP long. Base pair. BP means base pair, guys. And contains single H1 histone proteins. Generally, DNA makes 2 complete turns around the histone octamer, and these 2 turns, 146 base pairs long, are sealed off by H1 protein molecule. The association between minus negative charged DNA positively packed histones allows for meaningful DNA packaging inside the nucleus. Number of nucleosomes in human is 3 in 10 to the power 10. So by this we have completed our topic, and I'm gonna share some new questions in the next video about this topic. So guys, I'm taking off. So guys, we'll meet in the next class. Like, share, comment, and subscribe. See you later.